Our next talk is by Kutluk Oktay, DNA repair, BRCA gene function, telomeres, and oocyte aging. Hey, Kutluk.
Thank you. Good morning. And David and I have been chasing each other on lecture circuits. We were in Spain like last week, following the same sequence. Hola. Again, thank you for the invitation and Dr. Zhang and New Hope Fertility making me part of this exciting event. What I want to do today is to go over another plausible mechanism of oocyte aging, translational work that led to some of the mechanistic findings that may explain some of the changes that come with age to oocytes. So in this talk, I will go over that DNA repair mechanism of aging theory, and then also review the impact of BRCA mutations and ovarian reserve in fertility. And if I have time, I'll give you some also on the male side of things. So even though we have a high-level audience here, we have some residents, fellows, perhaps students, so I just want to go over some of the basics that though in the fetal life there are very few primordial germ cells, the number of those cells multiply through mitosis to multiple millions. Some are mid-gestation, and there's a rapid loss of these follicles at birth, and women are born with about 1 million or so primordial follicles. The prevailing opinion that these follicles, primordial follicles, are like Sleeping Beauty. They just lie there for many decades, kind of in an inactive state, just passively subjected to environmental and other damage, while their oocytes are resting in prophase 1. Well, a little bit of diversion here. Early years of my training, I had the opportunity to work with a ovarian physiologist, done some important work in ovarian aging and in general aging, Jim Nelson. And later on, I went to England to work with one of his collaborators, Roger Gosden, and these two had collaborated. Fatty Gosden, who's a mathematician, and had done this work. What they'd done was to look at the number of primordial follicles in human ovaries from cadavers. And their observation was that there is a steady decline in number of follicles in the ovary up until age 37 or so. And there's, depending on which model you look at, there is a change around age 37 or 38. Now, in this very wasteful process where you're only ovulating for 500 oocytes, but the rest are becoming atretic, if you could actually control this decline in rate of follicle loss, you could extend reproductive life. And this has become my actually main research as a fellow, and then oocyte ovarian aging in general. And every other phenomenon in reproduction that comes with aging follows the same pattern. Natural fertility rates also decline sharply after similar age 37. IVF success rates start dropping sharply after 37 or so. And the percentage of euploid embryos or oocytes also. So there is something that happens around that time that somehow the paradigm changes. Now, in parallel, I've spent close to 20 years now working on fertility preservation, and we've developed some approaches in answering various scenarios with cancer patients. So one of them was to use aromatase inhibitors to stimulate women with breast cancer prior to chemotherapy. And while doing that, we have encountered many women with BRCA mutations. So what we observed was that in these women, especially with BRCA1 mutations, they produce fewer oocytes than those untested or negative. So this led us to ask some questions. BRCA1 and as well as 2, these are DNA double-strand break repair genes. Even though the mutations are, let me go Yeah. There you go. Even though the mutations are about 1 in 1,000 in general population, it could be much more common in certain ethnic populations. But DNA double-strand breaks are very serious insults to the genome. As a matter of fact, we all are subjected to this kind of damage constantly. There are about 1 million of those happen every day, and they're repaired. And as opposed to single-strand breaks, which could be easily repaired because you have your sister strand there, which could be used as a template. When you have the both strands broken, that requires a much more precise repair mechanism. And several mechanisms evolved in that. And one that fits oocyte the best is homologous recombination, which uses the homologous chromosomes to repair these double-strand breaks. So to establish that a complex mechanism evolved called homologous recombination or DNA double-strand repair pathway. And as you could see, BRCA1 is central in that repair. BRCA2 is more peripherally involved, but there's a whole sort of other genes that are involved in here. Just to briefly go over that, when there's a double-strand DNA break, H2X is phosphorylated, histone protein, and attracted to all these double-strand break sites. You can actually visualize these molecules and quantify breaks in a one-to-one fashion. So we'll be using this to show DNA damage changes. And in response to these breaks, various proteins are involved. For example, MRN complex, it just binds to these sites, serves as a platform where the other repair proteins can act and repair DNA. BRCA1 is involved in that process as well as in checkpoint and sensing damage. If this cannot be repaired, the outcomes are either cell cycle arrest, what we call cell senescence, or cell death. So this ensures that cells do not pass genetically damaged material on to next lineage. So then, putting these observations together, we asked the question whether DNA DSP repair and related genes could be critical in oocyte aging. And could the decline in DNA double-strand break repair efficiency be responsible for that age-related decline in ovarian reserve and change in other reproductive outcomes past age 37? So this led to this work that we published that I will summarize, and then I will add some unpublished data to this. This was a translational work. We've started with mouse and paralleled it into human tissue and then went to patients. So I'm going to give you mouse and human parallel because I think it's important to show the validity of the data. So when we looked at mice and their primordial follicles or their GV oocytes and looked at the percentage of gamma H2X, again, from my earlier slide, gamma H2X is a marker of double-strand DNA breaks. So if you look at these mice that are young versus old, there's a significant increase in double-strand breaks in their primordial follicle oocytes, and you see the same thing in mature oocytes. And the same true for humans in human primordial follicles from young. So these patients are separated by about a couple of decades, and there's tremendous increase in DNA damage in these oocytes, both in primordial follicles and GV oocytes. These GV oocytes were obtained from IVF cycles. These are discarded GV oocytes which were not in vitro matured. This is actually a telling picture. We're looking at mouse oocytes, and on the left you have the young oocytes, and on the right you have the old oocytes. And the pink is marking double-strand DNA breaks. That's gamma H2X. So you can see that what an equivalent of a couple of decades of aging in humans, equivalent in mouse, could do, where you go from oocyte without barely having any pink to all the way to severely scarred oocytes here. So clearly, there's a lot happening in terms of DNA integrity with aging to these oocytes. So then we ask this question. If this is happening, is that due to the fact that oocytes' ability to repair DNA is going down? So to answer that question, we collaborated with Dr. Goswami, who's at Yeshiva, who's a phenomenal molecular biologist, especially in single-cell analysis. So using, again, discarded human oocytes from IVF cycles, we did a single-cell real-time PCR looking at key double-strand DNA repair genes and correlated that with age. So to, again, remind you of the pathway, BRCA1, our key interest, plays multiple roles in this repair. BRCA2 is more on the homologous recombination side, so we studied these. ATM, sort of the orchestrator of all this process, so we studied that. As part of this damage sensing complex, MRE11, we studied that. And RAD51, again, critical in homologous recombination, we studied that as well. So what we found was interesting. So these are oocytes from patients. You're seeing that they're ages 23 to 41, 24 patients. These are the data from single oocytes, not pooled data, and relative expression of these genes in real-time PCR. So what we are finding is that there's a significant age-related decline in BRCA1 gene expression. So as oocytes get older, or as women get older, their DNA repairability in their oocytes go down with BRCA1. And we saw the same thing with MRE11, RAD51, and ATM, but we did not see that statistically significant trend with BRCA2. These data were also confirmed with protein expression under confocal microscopy. Then we asked this question. So I showed you this decay curve with the primordial follicles and the acceleration after 37. Is it possible that decline in DNA repairability increases, also accelerates after age 37? So when we broke this down to below age 35 equal or below or above 36 and looked at the slope of the curve, what we see is actually with BRCA1 that there's not such a change. It's more steady. But when you look at ATM, MRE11, RAD51, after age 36, there's actually a sharper decline. And interestingly, BRCA2, I told you that we didn't find any significant correlation with age and BRCA2. But once you look at past age 36, there's a significant trend. So this actually makes sense. If you think about what happens with cancer risk, BRCA1-related cancer risks are earlier. They tend to happen earlier. And with BRCA2, it's later. So I think what's happening is that the decline in BRCA2 function is much later, and that's sort of at the fringes of reproductive lifespan. So that's why we don't see as much impact with BRCA2. So that kind of fits this decay curve, what we see happening with the majority of the DNA repair genes. Since then, there have been several other animal studies confirming these findings. For example, in rat, it's been shown that BRCA1 gene expression declines with age. And also in primates, in monkeys, there is increased double-strand breaks with age in oocytes, as well as declining BRCA1 expression with age. So the data from now 4 species, human, mouse, rat, and primates corroborate. So then we said if that's the case, how about we look at a transgenic mouse model and see what happens with the reproductive function of these animals. And we've obtained these mice from NIH. Now, if you knock both BRCA1 genes, these animals are not viable. It's a critical gene and they cannot survive. So we studied the homomutant for that. But BRCA2, you can have a homomutant, so we studied both homomutant and heterozygote for that. So what we found in BRCA1 mutant mice was identical to what we saw with patients. First of all, up here, you're seeing the oocyte yield after stimulation. With BRCA1, the mutant mice has much fewer oocytes, but not BRCA2. The number of pups after mating, also fewer with BRCA1, but not BRCA2. The BRCA1 mutant mice have fewer primordial follicles at 5 days of life and 4 months. And at birth, around 5 days, the damage in their oocytes, DNA damage, is similar to controls, but it increases with age. So they're more susceptible to accumulating damage. Again, we don't see that with BRCA2. It doesn't appear to be as critical. Then we said, if this is so important, these genes are so important, could we then manipulate the oocytes and make them more or less susceptible? So we did a series of experiments involving downregulating the genes as well as overexpressing, and then exposing to genotoxic stress in the form of hydrogen peroxide. So when we downregulated these genes, what we found was that in 3 different categories, one, what happens to double-strand DNA breaks, 2, apoptosis, and 3, survival in culture. Here is the non-injected control. This is the scrambled microRNA control, which is sham injection, sort of. And when you knock down BRCA1, MRE11, RAD51, or ATM, in about 16 hours, double-strand DNA breaks increase tremendously. So it's an acutely important process. Same way, the apoptotic oocyte number increases, and the oocyte survival severely declines. Then we said, well, what about the opposite? We took BRCA1, so we took some old oocytes. So if you expose old oocytes in culture to hydrogen peroxide, this is the survival we get. But the moment we inject them with BRCA1 cDNA and overexpress them, we bring them to the survival level of a young oocyte. So this is telling that, in theory, if I can upregulate your BRCA1 expression, maybe I could make your older oocytes like the younger oocytes. And then we said, well, if all this is true, then we should see some kind of diminished reserve in women with BRCA mutations. So it's different than age-related decline, where both alleles are losing their function with age. This is not exactly the same model as aging, but we should be able to see some kind of difference. So we studied, in collaboration with Memorial Sloan Kettering, we studied women with BRCA mutations who were diagnosed with breast cancer, and we looked at their AMH levels in prospective fashion and compared that to BRCA-negative women. So what we found was, again, consistent with what we saw with earlier data in mice. In women who had particularly BRCA1 mutations, serum AMH levels were significantly lower. But this did not reach significance in BRCA2 mutations. Again, we discussed why this might be the case. Now, since then, we've also obtained ovarian tissue from women with BRCA mutations in collaboration with Brigham and Women's Hospital. Dr. Wayne Lin, who was a fellow there and came to work in my research laboratory, we collaborated. They provided the BRCA ovaries from BRCA mutant patients. We provided control. And what we found was, again, similar to what we saw in rodents, is that the primordial follicle numbers were significantly lower in BRCA mutation ovaries from women with BRCA mutations compared to controls. And also, the percentage of damaged oocytes, double-strand DNA compromised oocytes, were significantly higher in the ovaries of women with BRCA mutations. Furthermore, if you looked at the age-related accumulation of DNA damage in these women's ovaries with BRCA mutations, there's a much faster accumulation of DNA damage in women with BRCA mutations compared to who did not have these mutations. So clearly, whichever angle you're looking at this from, you're arriving at the same conclusion. So how about the rest of the literature? Let's take a look at that. If you look at the major studies that look at BRCA mutations and menopausal age, majority of them will show you earlier menopause, especially for BRCA1 mutation carriers. Now, there was one study that looked at the percentage of women reaching menopause in women with BRCA mutations versus relative controls. But in that study, only 19% really reached menopause. And they didn't find a difference, but I'll show you in 2 slides later, they actually reversed their positions. In their follow-up paper, they criticized their own paper. And so this is now in the positive category. In terms of measuring AMH in BRCA mutations, again, majority of the studies are showing that there is a difference in favor of mostly BRCA1. There was one study that did not find difference, but it was a retrospective study on undefined controls, and they included polycystic ovary syndrome patients. They were not excluded. As you know, they skew the curve. But I think this most recent study kind of put an end to this discussion. This was the group that did this menopausal age study, where they looked at when they reached menopausal age. Then they said, well, let's study AMH levels in a large number of women. What they found was that in women with BRCA1 mutations, here's the BRCA negative, mutation negative patients, and this is the BRCA1 mutation positive patients. So you could see that with age, this gap widens around here. So they have significantly lower AMH levels, but they didn't see that in BRCA2 mutations. And now there are genome-wide association studies in large number of women, over 70,000 or so, looking at the genetic determinants of menopausal age at menopause. And what did they find? The most significant variants are within DNA repair, and majority of them are in the homologous recombination domain. So if you look at the list, those that are in bold are all double-strand break repair genes, and majority of them related to BRCA. So why then many of the studies that looked at fertility in BRCA mutation carriers did not find a significant difference? Well, here's the reason. First of all, there are two alleles, and when you have a mutation, you still have a healthy allele. Nothing is going to happen to you as long as you have your healthy allele. But as with age, the function of the other allele goes down, then problems occur, and you have breast ovarian cancer and increased sensitivity to environmental genotoxicants and accelerated oocyte death and earlier menopause. But what happens in cancer situation? If you have a very severe BRCA dysfunction, you either develop early cancer, you have chemo, or your ovaries are removed, or you have early prophylactic salpingo-oophorectomy. So nobody is going to be able to judge your ovarian function. And so if you look at the studies that only follow those that did not develop any cancer and they were able to reach early menopause, you're looking at a population with the least dysfunction of BRCA. So with that, of course, we're only seeing maybe 2 years worth of early menopause. They are early, go into menopause a couple of years earlier. So clinically speaking, we're not seeing this huge difference because we're only looking at the tip of the iceberg. We were able to observe what we observed with the fertility preservation patients because we were able to look at somewhere here. These patients normally would not have had a chance to reproduce or go through ovarian stimulation, but with aromatase inhibitors, we were able to open a window into that. Just quickly about egg quality, because I think that's as if not more important than what happens to numbers. But if you knock BRCA gene, you actually cause spindle misalignment, and it has a strict relationship to tubulin function. Moreover, ATM repair is closely involved in cohesin function. Subunit of cohesin is actually a member of ATM double-strand repair break pathway. So there's an intimate relationship there. Well, Bob Edwards actually made a very interesting observation in 1968 that looking at two different species of mice, what he found was that with aging, the number of chiasmata declined in these mice. So the homologous recombination pathway actually also maintains the chiasmatic relationship. So is it possible that this is a reflection of declining double-strand DNA break, which results in less cohesion between chromosomes and predisposing them to aneuploidy? And finally, to tie it to Dr. Keefe's theory of telomeres in aging, as a matter of fact, if you knock BRCA, you result in telomere shortening. So as you would see from all these talks, that all these events are related. So in the end, we've come up with this hypothesis that could potentially explain what happens to ovarian reserve and egg quality with age. That is, we're going to come to him. That is that with age, there's a decline in double-strand DNA repair and BRCA function that leads to accumulation of lethal double-strand breaks in oocytes. And if they cannot be repaired, then these oocytes are eliminated through these check mechanisms. And since this decline is more significant after age 37 or so, you're seeing that accelerate after a certain age. Again, because of BRCAs and other DNA double-strand repair proteins involvement in meiotic function, this may also explain increased risk of aneuploidy. Obviously, I told you that telomere dysfunction may tie into this, and Dr. Keefe will tell us about this later. With that, what about men? I don't have time to talk about this, but we're actually finding exactly parallel findings. This is my PhD student now, graduated, and there's a lot more data behind this slide, but I don't have time to show you that in men, we're finding the same thing, decline in DNA repair in sperm, as well as those with BRCA mutations. We're seeing similar findings. Perhaps a future talk could cover that. With that, to summarize, it appears that DNA double-strand break repair mechanisms, especially relating to BRCA1, play a critical role in ovarian aging, and there may be a possible similar role in sperm. But what we need to do is work on upstream mechanisms, and which may actually tie all these different theories around there under one unified theory. And we're also working on targeted treatments to enhance DNA repair. As a matter of fact, we have some preliminary data I can share now that with a certain agent, we're seeing that we can enhance double-strand DNA repair in oocytes. So if we could do that, perhaps could we find a treatment to enhance oocyte quality? With that, I'm going to get out of the way and show you the people who truly did work, specifically Dr. Shiny Titus, who is the senior postdoctoral fellow in my lab, and Dr. Stobeski is the PhD student who graduated and now postdoc elsewhere. Thank you again for your invitation.